I'm Ilia. I play a role in helping to collect the plants that are needed for the Darwin Tree of Life project. And I'm here today to collect more plants from this particular wonderful area, the Lizard in Cornwall, which has got a fantastic diversity of plants, some of which occur nowhere else in Britain. So all around you, you've got wonderful species that will really contribute to the Darwin Tree of Life project. So we found some really interesting species, including, for example, Erica vagans, the Cornish heath, which is a particular speciality of this area, so that was really good to get that one. We also collected some other very rare species, such as the wild asparagus, a close relative of the edible asparagus that we eat, and that's a crop wild relative, so who knows what information it may contain in its genome to help us to perhaps breed more resilient asparagus as we undergo climate change. So when we come to a plant we want to collect, the first thing we do is to, first of all, of course, check that there's sufficient number of plants that we don't put that species at danger of extinction. But once we've confirmed that, then what we want to do is to, first of all, we collect a sample for the DNA sequencing, which will ultimately get sent to the Wellcome Sanger Institute. And for that one, we need to collect really nice, fresh, healthy material, which we put into tubes, which are all barcoded, and then they get frozen very rapidly into dry ice so that instantly the DNA is preserved, which will give us the high quality DNA that we need for building the genomes for these platinum grade chromosome level genome assemblies that the Darwin Tree of Life is going to produce. So that's for the actual DNA sequencing, but in addition to that, we also collect a sample in order to estimate its genome size, because one thing about plants is that the genome size, the amount of DNA in the genome, varies over 2,400-fold within the different plant species. And that, of course, is really important because when you're sequencing a genome, you need to know how long is the genome. So you know how long is it going to take, how much data do you need, how much is it going to cost. So we collect a sample, a fresh leaf sample, which we will put into a plastic bag to keep it moist, and when we get back to Kew, we will estimate the genome size of that species. Then on top of that, we collect a sample for DNA barcoding. This is a check to make sure that we have indeed correctly identified the plant species that we think we've collected. It gets sent up to Edinburgh, where they're doing the barcoding for all the plants that are collected as part of the Darwin Tree of Life. In addition to that, we also collect a herbarium voucher. So this is our permanent record of the plant material. So if anybody has any doubt and wants to come back and look at it later, we have a pressed plant of the species that we've collected, and we make two of those because one will go to the Kew Herbarium and one will go to the herbarium in the Royal Botanic Gardens, Edinburgh. One real advantage of coming here is that we're working with the Botanical Society of Britain and Ireland, the BSBI, together with the Botanical Cornwall group. They have great local knowledge and expertise of where the particular plants that we're so keen to collect are. So they've been really helpful, enabling us to fast track the discovery and the collection of species in this area. We had a great group of people. There were 10 people who signed up to be part of this collecting trip. They were, I think, all local from Cornwall, and they were all very interested in the flora, and we worked with them to collect samples so that they are as much engaged with the plants that we collected today as we all are as well.